So on the behalf of Learn to Upgrade team, I would like to welcome you all for this session. In this session, I will be talking about basically the introductory industrial biotechnology. And then we will be having a short discussion about what are the job roles, what are the career opportunities, and what is the scope if you want to enter or you want to work in this field of industrial biotechnology. Okay. So let me introduce myself. I am Dr. Anshu. I have done my MTech PhD from IIT Kharagpur. I'm NEET and CSIR JRF qualified. And so let's start. Okay. If you look at, I'm audible. Someone has written your voice is not audible. I'm audible to all of you. Okay, fine. Those who are facing the issue of voice, kindly rejoin once again. I think the problem will be resolved. Okay. So in present scenario, if we look at, most of there are chemical processes, or we can say they are more of chemical processing industry are there. Okay. But the major challenge, what chemical industries or the researchers or the chemical engineers are facing today is that they need to you can say derive or design, okay, a process which is sustainable. Sustainable in terms of what? Means it should be, you can say the process should respect the environment. The process should improve the quality of life. The process should be such that the product is competitive in the market. At the same time, there should be a minimization of the hazardous waste. These are very common terminologies. We always hear about that. The chemical processing generating a lot of waste, it is hard. But to design a certain process, it takes a lot of effort. Okay. And this industrial biotechnology approach, where we are designing a whole process in a sustainable manner by using the concept of biotechnology. Biotechnology, you all know. If I say all of you to define a biotechnology, you will be giving a very brief definition about it. Okay. So if I say, ask you, what is a biotechnology? Can you define? Because most of you, the students, will be of biotechnology background. Okay. So can you define what is a biotechnology in a simpler term? Any of you, just define biotechnology.
Biotechnology is the study where we apply technology to give a better outcome of a biological process.
Right. Okay. So someone have mentioned about that it is a technique, or we can say it is a set of tools. Okay. Set of tools where we are modifying a certain living organisms. Okay. Either we are modifying or we are making them to adapt according to our desired output. Okay. And these living organisms can be anything. You can use a microbial source, you can use a plant source, you can use an animal cell. Okay. Even insect cells are nowadays are used. So there are a lot of range of living organisms which can be used to get a desired end product. Okay. Desired end product where we are focusing on bioproduct. Okay, so what exactly we are going to derive? We are going to derive a bioproduct. So before starting, before choosing any or setting any industry, what you need to target? You need to target what exactly is your end product. Means you have to fix your end product. Based on that end product, you are going to choose the source from where you can get that bioproduct. After choosing the source, the next step will be your bioprocessing. How you are going to decide this bioprocess design. Okay. This is a simple biotechnological process. Biotechnological process is advantageous in comparison to chemical process in terms of bio-waste utilization, or we can say recycling, because biotechnology is developing a sustainable type of process where it is focusing on the process and the production system. This is your production system. You are focusing on the process where you are saving the cost, process where you are making it more profitable in comparison to the chemical process, and as well, you are utilizing this bio-waste and recycling it. So what you are exactly doing, you are increasing the eco-efficiency, you are decreasing the level of pollution, and at the same time, the amount of energy. If you compare an industrial biotechnological process and a chemical process, you can see there is a lot of energy. The chemical processes are more of energy intensive in comparison to the industrial biotechnological process. And at the same time, the output, the product which we get, they are most of the time, they are not biodegradable. Okay, so they are hampering the ecosystem, or we can say they are hampering the environment. Okay, so in all this perspective, we can say that using the concept of biotechnology at the industrial scale, we are developing products which are better performing, which are more durable, and which don't persist after their useful life. Okay, so why their name is given a bioproduct? Because in the process, we are targeting to develop a product which is biodegradable in nature. Okay, so they don't persist after the useful life. They being less toxic. Sometimes these bioproducts can be reusable also. And at the same time, the waste which is being generated during this bioprocessing using the biotechnological approach, we are recycling it. So we are developing a concept of zero waste. We are developing a concept of zero waste here. So if we combine all these advantages which are related to industrial biotechnology is becoming one of the most fastest growing sector in India. Okay, if we look at today's scenario in India, there are industry which have more than 2,700 biotech startups are here. If we talk about there are more than 2,500 biotech companies are there, and in the time we can see that India will be becoming the one of the country which will be adapting this industrial biotechnological concept, because now the focus of the researchers is more towards your developing an eco-efficient process. Okay, so how this industrial biotechnology has been opening a better avenue in comparison to certain chemical processes. I will give you an example. Okay, BASF is one of the, you can say, company which is focusing on the food and nutrition aspects. Okay, and if we talk about this company has produced a vitamin B2, okay, which is a riboflavin, in less than in a single step in comparison to the 8-step chemical process. So here you can compare that just using a fermentation technology step, this BASF bio-based fermentation processing technique, this company has derived a vitamin B2, which is of the highest and the purest quality for its various applications in the dietary supplements and the food industry. Okay, and if we talk about the eco-efficiency of this approach, they have found that if they are going through this industrial biotechnological approach, the carbon dioxide emission has been reduced by 30%. Okay, and similarly, the waste reduction is by 95%. So just compare the chemical process, the effect it is giving to our environment. At the same time, using the techniques of biotechnological at the industrial scale, we are going to meet the demand in an eco-friendly manner. If we talk about the DSM is a company of is a global company, Royal DSM it is normally named as, and it is led by, its company works in the area of health, nutrition and bioscience. And the prime aim of this company is to improve the health of the human body system. We all know this cephaloxime. Cephaloxime is an antibiotic. This antibiotic belongs to the group of cephalosporin antibiotics, and it works on this bacteria because it's antibiotics. So basically, it will be working on the bacteria. And this cephaloxime is used in the case of pneumonia and certain bacterial infection in the case of your urine, different body systems like skin, ear, bone infection, UTI. In all of these cases, this antibiotic can be used. So this DSM company, which is used, traditionally they are used to make this cephaloxime using a 10-step chemical synthesis. Okay, they were using a chemical process to design the cephaloxime. But when they designed a process, they designed a bioprocess where they were using a combinations of the fermentation and enzymatic. I hope you will be having a basic knowledge of what is fermentation and what is an enzymatic reaction. Do you know what is fermentation and what is enzymatic reaction? Do you all have a basic knowledge of what is fermentation, what is enzymatic reaction? Okay, so what they are doing, they are using these combinations where they are using this concoctions where first they are going for the fermentations and then they are using enzymatic reactions to develop the cephaloxines. Okay, and if you compare in terms of your energy consumption, the energy intensiveness which the chemical processes are using and the materials, the raw materials which they are using to produce this cephaloxine, the cost is reduced by 65%. At the same time, energy consumption has been reduced by 50%. So just imagine the processes, the 10-step chemical synthesis process or the 8-step chemical processes can be reduced to 1 or 2 steps depending upon the process designing, a bioprocess designing, and choosing the efficient organism or its component to get the desired end product. So if you see in the BSF also, what they have fixed that they have to get a vitamin B2. In DSM has also fixed that they have to get a cephaloxime. So first they have fixed the bioproduct, okay? Then they are going to design a bioprocess, okay? So initially, if you're working in this area, your end product should always be fixed. It is not that you are designing a bioprocess and during the bioprocess, you are thinking that this will be produced. No, you have to target the end product. And through that only you have to move or you have to design your bioprocess, okay? Similarly, Novo Enzyme, you will all be well familiar with this company. This is Novo Enzymes. This company works with the enzyme sector. So this company, for you can see a lot of industries use, you can say harsh chemicals, okay, to wash off the dyes, okay? So in this scenario, they are not only discarding this dyes into the, you can say our water system, or we can say into the environment. Apart from that, these chemicals can be carcinogenic also. A lot of times the dyes are known for their carcinogenicity, okay? So what is normally the Novozyme came as a open or you can say derived enzyme-based process where they were not only focusing on that removal of the, you can say, dyes, but they were also focused that this when you are working with an enzyme, the whole process become water, you can say, this water intensive process, which normally the chemical textile industry use for their washing, by using the enzymes, the drop in the water requirement was 60%, okay? 60% drop in the emission to water was noticed. At the same time, the demand of the energy also decreased by 25%. So by using the enzymatic process, you're not only giving a better bioproduct, but you're also decreasing the lot of hazardous waste which are getting generated during the process of this chemical process. Apart from that, cosmetic and perfumes are, you can see, have topped with $15 billion in 2019, where they have focused on using the bio-based resources. Like for the nail polish, the acetone is normally used. Now, this acetone has been produced by this fermenting plant sugars. And apart from that, the palmitic acid that gives your hair that glossy shine, this has also been produced from the microorganisms. And questions I will be answering at the last. So just keep your questions with yourself. Just a session will be completed, then I will be answering each of your queries. Okay. So cosmetic and perfume industries are now focusing, because everyone is nowadays more oriented towards getting this bio-based products. So that's why even cosmetic and perfume industries are utilizing this. Similarly, the food additive and nutritional supplements like omega-3 fatty acids, these you normally, the targets were fish. From the fish, they derive this fish oil with omega-3 fatty acid. But nowadays, there are a lot of algae from where we can directly grow them at the higher level, you can say at a higher production level. And from there, we can go for the purification process, optimization, and then we can get this algae oil with better omega-3 fatty acids. Okay. Similarly, the plastics and other materials, biodegradable materials like cellulose derivative, PHA, which is being derived from bacteria, polylactic acid. There are certain bacteria which produce the lactic acid. From there, you can go for this polymerization and you can get the polylactic acid. Starch blends combining starch with the lactic acid. So there's a different ways of developing this bioplastics. Ecolife is a firm which is working in the Chennai, has using this concept of bioplastic for industry packaging. And the most important sector of the industrial biotechnology is a bioenergy sector. Okay, bioenergy sector is the emerging sector. You all know every country in the world is now nowadays focusing on the blending. Okay, 20% blending of ethanol or blending of biodiesel is one of the area of where they are achieving the sustainability by using the ethanol or the diesel, which is produced by, you can say, microbes. So bioethanol, you all know, and the biodiesel. In India also, the government of India has proposed that by 2030, there should be a proper blending of ethanol in petrol and blending of biodiesel in the diesel at 10%, 20%, and 5%, so that we can reduce the emission and at the same time meet the efficiency of the different types of, you can say, vehicle also. So all these, which I was talking about, the bioproducts. So bioproducts are products which are made with a certain component of biological or renewable material. And this bioproducts can be produced by your bioprocessing methods. So if I divide this bioproducts into different segments, you can get these can be biopolymers, these can be biochemicals like your organic acids, alcohols, aromatic, biodyes. There are different types of natural dyes which can be produced. Apart from that, you can get bioadhesives also. Biomedicine, biomedicine is also more of, you can say, emerging area of industrial biotechnology. And in India, the major portion of this industrial biotechnology is covered by this biomedicine portions. Biopharmaceuticals covers the major portion in India. And then nowadays, biopesticides are also being used, because a lot of pesticides and herbicides which are being used in India are reported to be mutagenic or carcinogenic. So there's a lot of, you can say, discussions and debate is going on. So in such scenario, biopesticides can be a better option in comparison to that of chemical pesticides. So bioproducts can be divided depending upon their use, okay? And apart from that, how much they are produced. For example, this type of products like lactic acid, citric acids, these products are normally used in a very high volume. Okay, so when they are used in a very high volume, they are produced also in a very high amount. So in this case, type of lactic acid and citric acid, normally the process is very high yielding. Yield means lot of means your productivity is more. Okay, productivity is more with the time. So there is a lot of high yield is there, high rate of conversion is there, high titer value is there, and they are required in high amount. So when something is produced in very high amount, its value is very low, means it will be very of low cost. Okay, so all those products which are produced in very high volume, they come under this high volume, low value products. Okay, some are medium volume and medium value products, means they are produced in very you can say more than the low quality, but their you can say their requirement is also medium. Okay, so normally these antibiotics, single cell proteins, vitamins, they comes under this medium volume and medium value products. Okay, now the most important type of bioproducts are that which are produced in very less amount. So when something is produced in very less amount, its purification cost will be very high, because you have to use a lot of, or you have to optimize this purification step so that you can get a high yield at the same time high tetra value. So that's why, so when something is low, its cost will be high. So usually these are required in very high amount. Most of the vaccines, insulins, antibiotics belongs to this low volume, high value products. Okay, and the major emphasis when we work with this low volume, high value products, because if you see vaccines, these insulins and antibodies, the major prime you can say parameters which need to be considered is that stability. They should be highly stable. And if you are using them for expression, obviously you have to look out at the productivity, at how much productivity is there, that your microbes or the cells or the organs which you are using for this production, how well they are expressing those proteins. And apart from that, you have to always keep in that mind that your production cost should be less. Okay. Now, this was all about biotechnology. So when we are talking about this industrial biotechnology, we are majorly talking about the scaling up. I think most of you have already been working in the lab. Okay. I think, do you all have experience of a lab? Do you have worked in the lab, biotechnological lab? Okay, so all of you have worked in the lab. Okay, so when we are talking about the lab, okay, so what was the first step? What was the first step? Your teacher or your madam or your sir, anyone gives you an objective. Okay, they give you an objective. Obviously, in your lab experiment, they give you an objective like this you have to produce, this you have to enhance the productivity. They give you an objective. Okay, so once they give you an objective, then you start designing your process. Okay, so when you start designing a process, when you're working with this biotechnology, what you will be doing, you will be either choosing a microbes. Let us take an example that you want to produce an enzyme. Okay, so what you will be doing, your teachers give you an example that I want this enzyme to be produced. Okay, so your target is now enzyme which you're going to produce. Now, next step will be that you will be choosing any living organism. Okay, like you will be using a microbe if you want a microbial source of enzyme. If you want a plant source of enzymes, you will be going for a plant source of enzymes. So you'll be taking a plant cells. Now, if during this process, when you have chosen it, next step, what it will be? Now you will be modifying your microbes. You will be checking it that it is producing that enzyme or not, or you will be adapting your microbe in such a way that it gives you more of enzyme. Okay. So similarly, this you work at the lab scale. Okay. But when we are working with industrial biotechnology, we are working with scaling up. We are not talking about the lab scale today. Today, we are talking about a full commercial scale production of that enzyme, which you have worked at the lab scale. Okay. So the scaling up is increasing something which you have done in a very small amount to an amount which can be commercialized, which can be directly enter into the market. So how can you scale up something? How can you enhance the productivity? Or how can you make something to go from a lab to a total from full commercial scale? Either you can focus on the yield, means either you are focusing on increasing the yield in a short time. See, time is very important. In every process, the time is a very critical point. Because if something is taking a lot of time, obviously it is going to affect your production cost. So limited time, or you can say defined starting time, defined ending point will give you a better yield. That is your best scaling up. Okay. Or you can go for batch size, means you have defined a set of point or defined an end point, and now you are just replicating it in a batch form, okay, in a batch bioreactor. So batch size is also one of the way where you are enhancing or getting a better production. And there you are increasing your process at the full commercial scale. See, something cannot be duplicated directly to the full commercial scale. There are certain steps. Each steps need to be considered. Okay. So lab scale is also known as, this lab scale is also known as bench scale. Okay. So the bench scale or the lab scale systems are importantly early stage tools. All the industry have their R&D sections which will work on their lab scale, or you can say a bench scale, where they will be assessing each of the parameters at 0.5 to 10 liter fermenter. Okay. Once they will get a better yield, or you can say a better production, they will be moving it to the pilot scale. Okay. Pilot scale is the scale which perfectly mimics your industrial scale production. Here, the quantity of your bioreactor is going to increase from 10 to 100 liter production unit. And similarly, the demo scale is also a very important, plays a very important, you can say, role, because your production unit is now reaching to 100 to 10,000 liter. That this demo scale differs from your pilot scale in that it closely resembles, this demo scale closely resembles to that of full commercial scale. Okay. And once your process is fully optimized at this demo scale and the pilot scale, then only the company is going to set up a full commercial plant. And always when the process is new, it is novel, means it is the first time process, then they choose this process of lab to pilot, pilot to demo, and demo to your this full commercial scale. But when just company has already set up your one unit and they want to set up another unit, they just follow the process where they replace this demo scale, because that process is already being optimized once at the full commercial scale. They just directly move from pilot scale to the full commercial scale. Okay. So lab scale, as I've told you, every company have their R&D section. So this is a discovery phase where you have a better opportunity to handle and carefully measure. But when you are working with the pilot plant, you are entering into the first window of the continuous processing, means your product will be produced, it will be purified, it will be packaged, and it will be checked for the quality. Okay. So all the performance tests should be done in the case of pilot plant if you are going for the full commercial production. So it acts as an advantage, because if sometime what happens, you have set up a full commercial plant, and sometime what happens, some unexpected problems take place. Suddenly your system, your productivity fails. Okay, so what will you do? That more than 10,000 liter, your full commercial plant has stopped. Imagine what will be the loss will be happening to our industry. So that's why the company prefer the pilot plant study, then they go for demo scale study, because if certain thing is going to affect the production system, then they can afford that changes at the pilot speed and demo scale. Okay, so these, with the increase in the scalability, there will be more of higher capital operating cost will be involved. Okay, and as a process engineer or your scaling up engineer, you have to take into the consideration that your capital and operating cost should not be exceeding of that of your product cost. So you have to maintain this capital and operating cost in such a manner that industry affords it. Okay. So if we just combine this all, what we have talked about the scaling up. So scaling is that lab you have done. Then you will be developing a plant flow chart, how you're working with the plant. Now you will be setting a bioreactor, and at each this step, there will be a role of a processing unit. That because just production is not our requirement. Our requirement is purification also. Our requirement is packaging also. Our requirement is that the waste which is generated should be utilized, properly treated before it is getting discarded into the environment. So this whole process, the scale up should be, process flow should be considering this processing, purification, packaging, and waste. And as an engineer, as a biotechnologist, as an industrial biotechnologist, you have to link each of the steps. You have to optimize each of the steps. You have to verify each of the steps. You have to do risk assessment at each of the steps so that your life cycle of your bioproduct should be maintained properly. Okay. So the parameters is that you focus on increasing the process intensity. Okay. Initially, you don't have to focus on the scaling up volume. You just have to increase the process intensity. Once your process intensity has been reached to the desired amount, then you go for the scaling up. So you don't start suddenly with the scaling up. You increase your production intensity and then you go for the scaling up. Because your prime aim is that you have to aim at that your production costs should never be higher than your substrate or your product cost. Okay. Apart from that, your yield should be your prime importance if you are working with your bioproduct. Okay. So how you can do this? You can do this by, just a second. How can you do this? You can do this by using 6 simple steps, or you can say this is a very simple steps, but they are very important, and you can say very complicated when you work at the larger scale. That when you are adjusting your formula, something which can be adjusted at lab scale is very easy, but when you are working at a large scale, adjustment of formula is very tedious, and you can say very you can say intensive, energy intensive process. So you have to take into consideration that what formula is working best at the large scale. Right equipment. What type of equipment you are going to use? Are you going for the continuous type of bioreactor, or you're going for the batch type of bioreactor? All these needs to be considered. Cleaning and sterilization, one of the most important steps when you're working with the bioproducts, because a lot of time, if there is a contamination, your yield is going to decrease because other microbes will be using your sources to grow, and your desired yield will be getting decreased. Identifying relevant parameter codes. You know, in the industry, all the parameters are automated. So you have to just choose which are the parameter codes which are highly relevant for your production system. Optimization, use the mathematical optimization techniques to optimize your processes. And if you need any changes, like if you want to work into the changes, you have to go for the changes in instrumentation and the diagnostic at the pilot and the demo scale, not at the full commercial scale. Okay. So just to give an example, You can choose a microbe, you have to adjust a formula in terms of a feedstock, and apart from your media, because your media will be using, because if you're using any living organism, initially they need to adapt. In that case, media should be there, the minimal media should be there. And normally industrial grade raw materials need to be validated whenever they are using for the large scale. For example, when you're using a feedstock from a different sources, so their chemical compositions will be different. So you need to, you can say, analyze all of these parameters, chemical composition, physical characteristics of your feedstock before going for your lab scale. Similarly, when we work in the lab, we use a reagent grade or lab grade, but the industry cannot afford this reagent grade and lab grade because these are very high in amounts. So normally industry prefer this bulk grade or technical grade for this commercial and industrial purpose. And this technical grade and the bulk grade have a different quality in comparison to the reagent grade or lab grade. So something, bacteria or the microbes which are going perfectly in your reagent and grade or lab grade, sometimes it can happen that they are not growing well in the technical grade. So you have to go for the adaptation or of your microbes or of your cells, then only you can move ahead. Similarly, select the right equipment. For example, cellulase is used for the bioethanol production, but this cellulase, when the cellulose is produced, it is act as your inhibiting agent. So in this case, you have to use the right equipment. Either you go for batch culture, fed batch culture, or the continuous culture. Depending upon your requirement, you can use the equipments. As I've told you, cleaning and sterilizations are very important, and the parameters which need to be considered are time, temperature, moisture, air removal, drying. These all need to be considered when you're working with the bioreactors. So identify relevant parameters, both the parameters which normally are considered in the case of biotechnological processes. Scaling up is temperature, pH, agitation, incubation time, media composition, shear stress. Normally, shear stress is a very important parameter which need to be considered. Foaming is one of the parameters which need to be considered. So these are certain different types of parameters which we need to analyze as a scale-up engineer or as a process scientist. All parameters need to be considered. And finally, you have to go for optimization. There you will be setting up, computing the value of the functions, and you will be getting or maximizing or minimizing depending upon your requirement. Okay. So I have talked a lot about industrial biotechnological advantages. It's environmental friendly, it is potentially sustainable, it is green economy, but certain drawbacks are always related with any process. And the drawbacks is that, as I've told you in starting, that adjust your formula. The complex chemical nature of the raw materials which you are going to use is a very, you can say, create a lot of problem when you are scaling up. Okay, so this is one of the drawbacks. Another drawback is that you need a lot of investment. Okay, a lot of time taking, it is a lot of investments are required. And why new investments are required? Because chemical industries are already working in the system, and if they want to install, they want to use this industrial biotechnological way of it, they have to set up a new reactor technology. They have to set up a new technology based on their catalyst. There is a lot of transportation and production costs are there related with your raw material transportation. And most of the time, if you see, the raw materials are usually of their source is food. So there's a lot of debate on this food and that this raw material can be a source of food. Like for example, bioethanol is there. USA is producing that bioethanol from corn, and Brazil is producing this from sugarcane. So corn and sugarcane are used as food. So there's a lot of debate is that if you are using this corn and sugarcane for the bioethanol production, what is an alternative there? So in that case, a lot of agriculture based are being used. So there is a lot of disadvantages also associated with industrial biotechnology, and there we as a biotechnologist or an industrial biotechnologist has to play a major role. And this will open a new avenue, and you can say a job prospects in a career opportunity in this sector. So There can be a lot of employers in this sector. You know, government organizations are there, HPCL, Indian Oil. They are working in the area of bioenergy sector. So if you're interested, if you have a experience in the bioenergy, you can apply for this. There are a lot of internships are offered by this Indian Oil HPCL. You can work there. You can work after your MTech and after your PhD also. Private organizations like Reliance Life Science, Hindustan Lever, they are also offering this R&D sections. There you can work, and they have their production unit also. Universities, you know, you can join as GRF and SRF, Junior Research Fellowship, and Senior continue or MS, Master of Science, Master of Technology, and PhD. You can also go for postdoc in the different sectors. Biotechnological companies are also there, like your Syngenta is there, so Monsanto is there, so you can work there. Pharmaceuticals like Biocon, Serum Institute of India, they are also working. Agriculture companies are there. Shanta Biotechniques are there. They are also working in different sectors. Apart from that, there are certain companies like Thermo Fisher, Biorate. They majorly focus on the purification part. So if you have more inclination toward downstream processing, you can and you have experience in this downstream processing, you can work in this Biorate or Thermo Fisher water organizations. They have this, you can say, they provide you with different gas chromatography, HPLC, and different columns. So you can work with them also if you have knowledge about the purification for proper downstream processing. Okay. What are the job roles which you will be getting offered in this industrial biotechnologies? Initially, you can join as a productive assistant. You can join as an assistant. After that, there can be opportunities like technical officers. Initially, will not be getting like this type of jobs. Initially, you will be entering and you will be having an experience, and then only you will be offered this type of jobs: technical officers, scale-up engineers, project managers, quality control executive, process development scientist, and your formulation as a R&D scientist. So this all will be depending upon your qualifications. If you are a BTech or master in microbiology, biotechnology, you have to go for certain certification course in this case of industrial biotechnology. And if you're having experience, hands-on experience on how fermentation kinetics is done, how downstream processing is done, you have handled a certain fermenter, then these will going to generally add benefit to your CV. Okay. And what are the job responsibilities which you will be mostly focus on is on the bioprocess design. So bioprocess design is a conceptual work, as I have told you, that is based on building, expanding, and retrofitting a process plant. And it basically depends upon two aspects. One is a process synthesis and another is a process analysis. Okay. So when we are talking about the bioprocess design as a different type of job roles which you will be getting, you will be either working in the process synthesis or you will be working in the process analysis, where you will be evaluating comparison of different process synthesis solutions. So every step at which you will be getting an output, you will have to analyze the process. And here comes the role of the quality control executive and the quality analysis. And if you're working the process synthesis part, you are going for the selection and arrangement of a different set of unit operations, okay, where you will be focusing on the cost and the quality. And here comes the role of process development scientist, formulation R&D scientist, or the scale-up engineer, or the technical officers, process engineers. So all these roles will be entering into the process synthesis, and all the quality control, all the risk assessment, all will be under this process analysis. Okay. So apart from that, you can get a teaching positions. You can work as a project coordinators. Different institutes, organizations have a projects which they get funding from DPT, DST, or your CSIR. There, they require certain project coordinators. So you can work there. You can join different institutes, like there's a TERI where you can work as a scientist positions, but this all depends upon your qualifications. It requires a very higher degree like MTech, PhD, and postdoctorate. So depending upon your qualification, you can work in this sector also. And nowadays the emerging sector is your editorial, where you can work as an editor in the different types of companies which are offering this, you can say, language editing and etc., where you can join. Okay. So with this, we have come to the end of our session. Thank you, everyone. I hope it was easy for you to understand. So thank you, everyone, once again for joining and being the part of this session. Do give your review on this link which I have shared. And if you have a question and query, kindly ask me. Okay. Thank you, Rashni. Thank you, Livia, for your response, Hibashri. Thank you, Megha. Do you have any questions someone asked? How to get an internship in the biotech industry? See, normally what happens, they open when you will checking in the companies, most of there are certain companies like similar to this Learn to Upgrade, they offer, give an opportunity to work with the biotech industry. So you can join through their internship program and then enter the biotech industries, or you can go directly to those industries and you can check for the internships. Okay, from there also you can get an option. If you're MSc microbiology, I would say better you go for a PhD, okay? Because directly you will not be getting a good job position in the biotech industries, or you can go for internships in the case of biotech industry. Then you can enter into this different sectors of biotechnology, industry biotechnology. Any questions more? Yeah, you see, if you regular check this different types of industries career opportunity, you can see they normally write the requirement, like what is the basic educational qualifications should be there. So depending upon your qualification, they will be choosing you for different types of job roles. See, since you're a BTech in a biotechnology, so you will not be much exposed to the, you can say, labs, okay? Because there is a lot of, you can say, courses are there. So I think you will not be much familiar with different types of lab working environment. So what I will say, you do a summer internship or your different types of 6 months project, which is normally is a part of this BTech curriculum. Try to get in a company, okay? So try to work in this companies where they are offering this 6 months some project or 1 year project. There you will be more exposed, okay? Or you can join different type of internships where they are helping you to get an hand-on experience in the biotech industries. And from there, you will be getting a better exposure. And after that, it will be helping you. It will be adding you to your CV. Okay, so that will make you different from others. Yeah, if you want to, you can see it depends upon the choice of the person. Like if he wants to work, if he wants to do his research or it's different type of programs of masters or master of science or master of technology, depending upon its choice and interest, he can or she can obviously go to abroad. There are a lot of fellowships which are being offered by different types of universities, foreign universities, for your this different type of studies. Like for your Germany, there is a one fellowship. I'm just missing out the name. The Germany offers that fellowship. Yeah, Anshu, that is a DAT fellowship where you can go for that. Then USA also offers. Then there is GSPS fellowship for Japan. Then there's a different type of fellowships offered by different type of you can say countries. Depend you just check out. You will be getting a different types of fellowship. Okay, you can also join in the project also. That certain universities in the abroad, they have the projects and they pay for you. When you join there, they pay for you that fellowships, so you can join to their project also. Okay, as a fellowship projects. Currently, I don't have a list of these fellowships, but if you will checking out that depending upon your basic educational qualification and in which domain you want to work out, you can just sort it down. You just check the fellowship for this on the Google. Also, you'll be getting a list of those fellowships. In which area you want to work, you just type it. You'll be getting a list of fellowships. In which semester of MSc we start applying for PhD? See, normally they prefer that your master's degree should be completed. So either in your last semester, that is your fourth semester, you can start applying. And if you write them that your semester is going to end by this session, by this time, they are going to consider it. If you request them, they normally consider it also. So it's not a much issue, but normally you should apply in your fourth semester, not before that. Any more questions are there?